பயாலஜி இன் தமிழ் கோர்வை சேனல் சார்பாக அனைவருக்கும் வணக்கம். என் பெயர் கண்ணதாசன். In this video, I am going to present classical genetics part 4. I gave the links for video 1, 2 and 3 in the description. I request all the students to please go through those videos so that you are able to understand the content of this video. The content of the video taken from chapter 2 Pure Science Botany 12th Standard State Board book, chapter 5 12th Standard CBSE book. The subtopics covered are 1. Binomial expression based on monohybrid cross, chromosomal theory of inheritance. Both the topics are not covered in this part of State Board book. In the same way, what is covered in the State Board book, Trihybrid cross 2.3.6, not covered in the CBSE book. So it will be very useful for both CBSE and State Board students as well as the NEET aspirants. I hope this video will be very useful for you. If you like this video, please press the like button. Share this video to all your friends. Comment on it. Place your doubt in the comment. Subscribe our Biology in Tamil Korvai channel. Press the bell icon to receive the notification. Now let us enter into this video. Before that, as usual, I request all the students to take their textbook, go to the particular page in your book, take the hints taking notebook and pen. Wherever you feel very important in every slide, you please take note of it. It will be very useful for you in the revision before the examination. Now let us enter into this video. Number 1, binomial expression based on monohybrid cross. Monohybrid cross. Mendel crossed pure breeding tall and dwarf pea plants to study the inheritance of one gene. Study the inheritance of one gene. So it is called as monohybrid cross. You know very well what is pure breeding variety. Pure breeding variety is the one which produce the same trait when it is self-pollinated for n number of generation. Then it is called as pure breeding, and usually it will have the homozygous genotype. You see here the tall with capital T, dwarf with small t. Then they are said to be the pure breeding variety. Only one type of gamete from each parent is produced. The pure breeding variety can produce only one type of gamete. The tall plant can produce only one type, capital T gamete. The same way, the dwarf plant can produce the small t gamete. F1 progeny plants, all of them were tall for Mendel with respect to the Pisum sativum. It is giving the appearance of one of its parent, that is tall parent. The tall parent, all the F1 plants were tall and dwarf not expressed. So this is the first F1 generation result Mendel obtained. Mendel then self-pollinated the F1 plants, and to his surprise, probably he did not expect that you would get dwarf. That is the reason why to his surprise found that in the filial 2 generation some of the offsprings were dwarf. The dwarf not expressed in F1 but expressed in F2. The character that was not seen in the F1 generation was now expressed. So selfing of the F1 plants, the two tall plants, selfing occurs. So they produced two different gametes, one with capital T and another one with small t. The same way from female side, one with capital T and another one with small t. Now let us see what is the result. The proportion of the plants that were dwarf were 1/4. 1/4 of the plants were dwarf and 3/4 of the plants were tall in the F2 generation. From the Punnett square, it is easily seen that 1/4 of the random fertilization, it is the random fertilization you see through Punnett square. capital T from male side can fertilize the capital T of the female gamete, and another 50% can fertilize the small t of the female gamete. So this is said to be the random fertilization. Okay, so 1/4 of the random fertilization led to capital T. It means the homozygous tall plant. 1 by 2, it means 50%, half of the total number of plants carrying the genotype capital T small t, 50%. Heterozygous dominant, it means heterozygous tall plant. And 1/4 with the genotype small t. So the phenotypic ratio become 3:1, genotypic ratio become 1:2:1. Though the F1 have the genotype of capital T small t, but the phenotypic character is tall. The F1 plants, all of them are tall, but they are having heterozygous genotype, heterozygous dominant genotype. Are you clear? Capital T and small t. At F2, 3/4 of the plants are tall phenotypically, and 25% of them are capital T, 50% of them are capital T small t, and 25% of them are small t. This is the F2 result. Genotypically 1:2:1 ratio, in which 50% the two stand for the heterozygous dominant, heterozygous tall plants. Right. This led to a phenotypic ratio of 3/4 tall, in which 1/4 with capital T plus 1 by 2 with capital T small t, and 1/4 again with small t. So 25% of them were dwarf. So the ratio become 3:1, and genotypic ratio, as I already told you, 1:2:1. The 1/4, that is 1/4 1 by 2 1 by 4 ratio of capital T is to capital T is to small t is mathematically condensable. This is the very important part of this video. Please concentrate more here. Mathematically condensable to the form of the binomial expression. Simply I say a plus b whole square. Okay, here it is given ax plus by whole square. That is the gamete bearing genes capital T or small t in equal frequency. So the two variables are present in equal frequency. What are the two variables? Capital T and small t is present in equal frequency of 1 by 2. The expression is expanded as given below. Now this expression a plus b whole square can be expanded. Let us see. Now you can see the binomial expression monohybrid cross result. A plus b whole square is equal to a square plus b square plus 2ab. If you are a mathematics student, you can understand it very easily. Right? So this is the expansion of the a plus b whole square. Now we are going to apply the equal frequency of capital T and small t here as a and b. A stand for you assume capital T and b stand for you assume small t. X and y stand for half and half. Okay, so 1 by 2 capital T plus 1 by 2 small t whole square is equal to, now we are multiplying 1 by 2 capital T plus 1 by 2 small t into same, because it is square, you know. So 1 by 2 capital T plus 1 by 2 small t. So 1 by 2 capital T multiplied with 1 by 2 capital T. So we get 1 by 4 capital T, it is equivalent to a square here, that is ax whole square. Okay, the same way now 1 by 2 capital T now crossed with 1 by 2 small t. 1 by 2 capital T crossed with 1 by 2 small t. So 1 by 4 capital T small t. Are you clear? So heterozygous dominant here. And in the next cross, 1 by 2 small t, this part we are taking, multiplied with this 1 by 2 capital T. 1 by 2 small t with 1 by 2 capital T. So 1 by 4 capital T small t. Right? So we have to add these two because both of them are with same genotype. So we are adding this 1 by 4 capital T small t plus 1 by 4 capital T small t. It is giving rise to 1 by 2 capital T small t. It is equivalent to 2ab. It means 2ax by. I think you would have understood this part very clearly. So we covered a square plus this 2ab we covered. Now let us go through the next cross. We are going to cross 1 by 2 small t with 1 by 2 small t, this one. So 1 by 2 small t, 1 by 2 small t is equal to 1 by 4 small t, that become b square. So the binomial expression goes like this: a plus b whole square, it means ax plus by whole square is equal to ax whole square plus by whole square plus 2ax by. Are you clear? So this is the binomial expression of the monohybrid cross result. Based on this, many questions can be asked for CBSE board student as well as all the NEET aspirant. You must be very thorough in this area. If you have any doubt, you please put it in the comment. I will answer for it. Now the CBSE textual question. It is given here. What do you think he, it means Mendel, would have got had he self-pollinated a tall F2 plant? Remember this F2 generation tall plants. What the result you would have got if he self-pollinated, if he had self-pollinated the F2 tall plants? This is the question, right? Now let us go for the answer. There are genotypically two tall plants. First, we have to understand it. We have to go by the genotype here. Whenever we see the phenotype, behind which how many genotypes are there, we have to take it into account. Phenotypically, though they are all tall plants, it means that is 75% of the F2 generation tall plants were there. Are you clear? Though the 75% of them were tall, in which what that is 25% of them are homozygous tall, rest of them are, it means 50% of the total number of plants I am talking about. Rest of the 50% heterozygous tall. Now, if he had self-pollinated what plant? So we have to give two possibilities. Possibility 1, capital T, cross capital T, self-pollination, homozygous tall plant. If he had done the self-pollination of this, you would have got 100% tall plant because there is no small t at all. Are you clear? Then possibility 2, capital T small t, it means heterozygous dominant is crossed with capital T small t, heterozygous dominant. Now it is equivalent to the F1 self-pollination, student, because in F1 generation we are crossing the two heterozygous dominant plants, self-pollination of two heterozygous dominant plant. Again we will be getting the same ratio, it means 3:1 ratio, in which 3 of them are tall and 1 will be the dwarf. So Mendel would have got either 100% tall plants if he had self-pollinated the F2 tall plants. This is one possibility. In another possibility, you would have got the ratio of 3:1 phenotypic ratio and genotypic ratio 1:2, 2:1. Are you clear? So this should be your result. Now we are going to discuss about chromosomal theory of inheritance. Chromosomal theory of inheritance. Mendel published his work on inheritance of characters in 1865, but for several reasons it remained unrecognized till 1900. So 35 years it was not discovered. His concepts did not come to light. What are the reasons for which Mendel's work remained unrecognized till 1900? So this part is purely discussed in CBSE book. This part I am telling, this part. Okay, in maybe some other part the state board book would have covered this information also. Reasons given. Firstly, communication was not easy. in those days as it is now, and his work could not be widely publicized. It is not known to all scientists of the world. Maybe it is publicized only in the local journals. Secondly, his concept of genes. He told the existence of factors, right, in Mendel words, as stable and discrete units that control the expression of traits and of the pair of alleles which did not blend. According to him, the alleles did not blend. When capital T and small t crossed, capital T expressed without any blending, they segregate from each other when the F1 plant produce the gametes. So no blending take place according to him with each other was not accepted by his contemporaries. Contemporaries also followed the continuous variation. Continuous variation. When we are crossing capital T, that is red and white. They were able to get pink in some other plant with some other character. So they say blending occurs. But according to Mendel, blending did not occur. So that is one of the reason why Mendel's work was not recognized. Thirdly, Mendel's approach of using mathematics to explain biological phenomena was totally new and unacceptable to many of the biologists of his time. They did not believe that mathematics is having any connection with biology. Today we have biostatistics, population genetics, it is full of mathematics only. Total statistical interpretation of various aspects of the population. Okay, so in those days they did not believe it. They did not believe the mathematics having connection with biology. But Mendel studied mathematics as one of his subject when he was studying in the University of Vienna. So he was able to interpret certain aspect of monohybrid cross and dihybrid cross with the help of mathematics. Okay. Finally, though Mendel's work suggested that factors, genes were discrete units, he could not provide any physical proof for the existence of factors. They asked, "You say factors, where are they?" He was not able to give any physical proof for the factors. And today we discovered the factors are nothing but genes. The genes can be seen only through the electron microscope, not by even the compound microscope. So he was very far advanced in his thinking and interpretation. So other scientists were not able to comprehend what he is coming to say in those days. So that is also one of the reasons why his work was not recognized by other scientists. So these are the reasons. So this can be asked in the NEET, which sentence correct for the Mendel's work not being accepted or recognized. Likewise, the questions can be asked. Rediscovery of Mendel's work. In 1900, three scientists, Hugo de Vries of Holland. The country names are given on the state board book but not in the CBSE book. Make it very clear. So CBSE students should take note of it here. Karl Correns of Germany and Erin von Schermach of Austria. So these are the three scientists independently rediscovered Mendel's result on the inheritance of characters. Also by this time, due to advancement in the microscopy that were taking place, scientists were able to carefully observe cell division. This led to the discovery of structures in the nucleus, structure inside the nucleus. What is the structure inside the nucleus? The main structure present inside the nucleus, the chromatin reticulum. It becomes chromosome during the cell division, appear to double and divide just before each cell division. The chromosome appear to double and divide just before each cell division. So the advancement in the microscopy led to the better understanding of the concepts of cell division. Chromosomes discovered. These were called chromosomes. Chromosomes means colored bodies. Chromo refers colored. Colored bodies as they were visualized by staining, by dyeing. By 1902, the chromosome movement like this during meiosis had been worked out. It means the chromosome coming together, pairing with each other, synapses take place, and also crossing over take place, and they separate from each other at the time of or during anaphase. All these were understood by 1902. Walter and Sutton, Walter Sutton and Theodore Boveri noted that the behavior of chromosome was parallel to the behavior of genes. Very interesting phenomenon. The behavior of chromosome was parallel to the behavior of genes, because you know very well the genes are located inside the chromosome. Each gene occupies its own loci, definite loci, specific loci inside the chromosome. So naturally there is some parallel behavior between the chromosome and genes, and used chromosome movement to explain Mendel's laws. So this made the scientists possible, made it easy for them to explain the Mendel's laws along with modern understanding. There is a modern scientific understanding of the cell division. Mitosis, equational division. Why it is equational division? Because the number of chromosomes present in the mother and daughter cells are equal, so it is equational division. And during meiosis, reduction division. Why it is the reduction division? The difference in the number of chromosomes with respect to the mother and daughter cells. During meiosis, the daughter cell consists of only n number of chromosomes, whereas the mother cell consists of that is a diploid number of chromosomes. Recall that you have studied the behavior of chromosome during mitosis, equational division, and during meiosis, reduction division. The important things to remember are that the chromosome as well as genes occur in pairs. The point to be noted here, there is some parallelism between the chromosome and genes. Number one, both occur in pairs. The two alleles of a gene pair are located on homologous sites of homologous chromosome. What are homologous chromosome? Identical chromosome, one derived from father and another one derived from mother, is called as homologous chromosome. So the alleles always present, the alleles, say example capital T, small t. Capital T and small t occupy the same gene loci of the homologous chromosome. They are present in the homologous chromosome. That is the reason why the genes and chromosome, the genes and chromosome behave in the same manner in certain aspect. And here you can see the meiosis and germ cell formation in a cell with 4 chromosomes. Here 4 chromosomes are present. This is one homologous pair and this is another homologous pair. During G2 phase, the duplication or doubling of chromosome take place, but still they are said to be the 2 chromosome, one pair, and this is one pair even after doubling, because of the presence of, you see, the centromere, only a pair of centromere and another pair of centromere. Only after the centromere division we consider the chromosome separated. Now meiosis 1, this yellow along with red gone to one pole, and orange along with green gone to another pole. Okay, so the two cells carrying haploid number of chromosome now, right? Now meiosis 2, the centromere, longitudinal splitting of centromere occurs, that led to the formation of, you see, the chromosome, haploid chromosome like this. Okay, each centromere carries only 2 arms, but here each centromere carries 4 arms. So longitudinal splitting occurs. So 4 cells are formed at the end of meiosis 2. From one cell, 4 cells are formed. Each consists of 2 chromosomes only. It started with 4 chromosomes. It means 2 homologous pair. Now it consists of 2 chromosomes only. Each cell consists of 2 chromosomes only. A comparison between the behavior of chromosome and genes given in the CBSE book. chromosome occur in pairs, gene also occur in pairs. Both the points are similar here. Second point, chromosomes segregate at the time of gamete formation such that only one of each pair is transmitted to a gamete. Genes, same point, segregate at gamete formation and only one of each pair is transmitted to a gamete. So both points are same. Where they differ from each other, the third point. You see the chromosome, independent pairs segregate independently of each other. Independent pair of chromosome segregate independently. First homologous pair, second homologous pair. Are you clear? When the chromosomes segregate, okay, they are independent. In each pair, they are independent. And you take genes, one pair segregate independently of another pair. One pair of genes present in one homologous chromosome, okay, it segregate independently of another pair of alleles present on the same homologous chromosome. Are you clear? So here they segregate independently. So second point refers genes and first point refers chromosome. From the CBSE book tabular column based question, right? Next, during anaphase, this is the anaphase, right? You can see the metaphase, all the chromosomes are arranged in a equatorial plane and they start moving at the time of anaphase. So during anaphase of meiosis 1, the two chromosome pairs can align at the metaphase plate. So this is the metaphase plate. Equatorial plane is called as metaphase plate. Independently of each other, can align at the metaphase plate independently of each other. To understand this, compare the chromosome of 4 different color in the coming slides. It is given in the CBSE book. Now this is the possibility 1. We now took two homologous pairs. First homologous pair with one orange, another one yellow. Second homologous pair, one with green and another one with red. Here the chromosome separate, segregate independently. That is a point here we are going to understand. Possibility 1, one long orange and green chromosome and long yellow and red, short red chromosome at the same pole. Are you clear? At the same pole. Okay, so possibility 1, orange and green is segregating together, orange and green segregating together and yellow and red segregating together. That is what we have to understand. These two go to one cell and these two go to another cell. Possibility 2, because it is independent in their segregation, one long orange and short red chromosome and long yellow and green chromosome at the same pole. Are you clear? But now they are going to segregate the orange with red, yellow with green. Previously, you see orange with green, yellow with red. But here independent in their segregation. So orange chromosome is segregating with red chromosome, yellow segregating with green chromosome. So what we are able to understand, one pair of chromosome, homologous pair, each chromosome of the homologous pair segregate independently of other set of homologous pair. So this is called as independent assortment. It is equivalent to independent assortment. We know very well that chromosome and gene behave in the same way. So if chromosome segregate independently, naturally gene also segregate independently. Okay, now Sutton and Boveri's work. Sutton and Boveri argued that the pairing and separation of a pair of chromosomes would lead to the segregation of a pair of factors they carried. Now the chromosome carry the factors, it means the genes. Are you clear? So the genes present here will be same here. Are you clear? So if the chromosome separate, alleles also separate. The set of alleles present here will segregate from the set of alleles present here. So this is what the work of Sutton and Boveri says. Sutton united the knowledge of chromosomal segregation with Mendelian principles. This is one of the wonderful work. He united the knowledge of chromosomal segregation that we are able to see under the microscope with the Mendelian principle he put forward without seeing the movement of chromosome under the microscope during cell division. Both now come true. It means the Mendelian principle, one he put forward without seeing the factors or chromosome under the microscope, come true. It is matching with the modern scientific concept of chromosomal separation. Both segregation and independent assortment come true and called it the chromosomal theory of inheritance. So this is what called as chromosomal theory of inheritance. Understood? The Sutton united the knowledge of chromosomal segregation with Mendelian principle. The same is called as chromosomal theory of inheritance. Work of Morgan and his colleagues. Following this synthesis of ideas, experimental verification of the chromosomal theory of inheritance by Thomas Hunt Morgan and his colleagues led to, so they started working on the chromosomal theory of inheritance with the help of some experiments, with the help of the Drosophila melanogaster, the fruit fly. The picture given, one is male and the other one is female. This led to the discovering the basis for the variation. What is the reason for variation occur through the sexual reproduction? They discovered. Morgan worked with the tiny fruit flies, Drosophila melanogaster, which were found very suitable for such studies. The Drosophila melanogaster found to be very suitable for such genetic studies or cytogenetic studies. Why? Why they are suitable for cytogenetic studies? The given, that is reasons given in the CBSE book. Reasons. Number one, they could be grown on simple synthetic medium in the laboratory. So it can be maintained very easily. Usually to have one laboratory animal, or if you select an animal or plant for your studies, it should be maintained in the laboratory. Suppose you take a banyan tree for your studies, is it possible for you to maintain in the laboratory or within the laboratory garden? Not possible. So this is one of the reasons they say. They complete their life cycle. They have their short life cycle in about 2 weeks. So many experiments can be repeated within a month. 2 experiments can be completed. Single mating could produce a large number of progeny flies. This is the third reason. Clear differentiation of the sexes. You see the male fly and female fly. Clear differentiation of the sexes. And they are easily distinguishable also. It has many types of hereditary variations that can be seen with low power microscope. The variation between the two flies can be easily studied under the that is low power microscope. That is the dissection microscope itself. We are able to see the variations between one may be with white eye and another one may be with red eye. We are able to distinguish the variation. Now we are going to discuss about the state board book content 2.3.6, which is not covered in CBSE book, Trihybrid Cross, right? The Trihybrid Cross, it means the study of 3 genes. In Monohybrid Cross, we learned that only 1 gene we studied. But here, we are going to study, it means Mendel studied 3 genes with the 3 pairs of alleles, right? So the Trihybrid Cross demonstrates the Mendel's law of applicable to the inheritance of multiple traits. Mendel law of segregation and independent assortment are also applicable to 3 pairs of contrasting characteristics called Trihybrid cross. A cross between homozygous parents that differ in 3 gene pairs. 3 gene pairs, it means allelic pairs, that is producing trihybrid, is called as Trihybrid cross. A self-fertilizing trihybrid plant forms 8 different gametes. Very easily we are able to understand how many gametes are produced by the F1 plants of Trihybrid cross. You just put the number 2 power 3, 2 power 3. Are you clear? 2 variation and number of genes studied 3. 2 power 3. So 2 into 2 into 2, 8. 8 gametes are produced by F1 generation. Suppose it is dihybrid cross, 2 power n. N is equal to 2. 2 into 2, 4 gametes. Very clear? Likewise, we are able to mathematically interpret and understand also. So 8 different gametes produce 64 different zygotes. 64 different zygotes. Okay, 8 into 8. There you just imagine the Punnett square of dihybrid cross. 4 into 4, 16 boxes. Am I correct? Here 8 gametes from anther, 8 gametes from that is pistil. Very clear? So 8 into 8, 64 possible zygotes can be produced. Very clear? In this combination of 3 single pair crosses operating together, it means you see capital T, capital T, capital Y, capital Y, capital R, capital R. Tall, yellow, round plant crossed with dwarf, green and wrinkled. Clear? So that is the reason why it is called as Trihybrid cross. Now with the help of this tabular column, we are able to understand the 3 character: height of the plant, seed color and seed shape. Traits: tall and dwarf for height of the plant, yellow and green for seed color, round and wrinkled for seed shape. And nature: tall is dominant, dwarf is recessive, yellow is dominant, green is recessive, round is dominant, wrinkled is recessive. And their genotypes given, which that is we already learnt. Now 8 types of gametes, how they are formed. 2 power n, I told you, n is equal to 3. So 2 into 2 into 2 is equal to 8. Okay, 3 refers the number of genes. Very clear? 2 refers number of variation. You imagine. Okay, capital T small t, capital Y small y, capital R small r. This is the genotype of the F1 plant. So we are writing it here, capital Y small y. This side capital T twice, small t twice. For each capital T and capital T, here capital R small r. So totally 8 gametes are formed. 8 gametes are. All of them are different from each other. Okay. And another way, you see capital T small t, it combined with any one of the capital Y small y, here capital Y small y, equal possibility we are giving for them to combine with other allele. Okay, other dominant or recessive allele. In the same way, this recessive can combine with dominant and recessive or recessive. In the same way, each combination of this again may combine with the third character dominant gene or the dominant allele or recessive allele. So we are getting the 8 gametes. This is called as fork-line method of finding out the gametes. 8 types of gametes from anther of F1 generation, self-pollinated with 8 types of gametes of pistil crossed through Punnett square. There will be 64 crosses, students, right? With phenotypically 8 different combinations. Phenotypically 8 different combination with the ratio 27 is to 8 different combination. You see 1, 2, 3, 4, 5, 6, 7, 8. 8 different combination in the ratio of 27 is to 9 is to 9 is to 9 is to 3 is to 3 is to 3 is to 1. So this is the trihybrid cross ratio, phenotypic ratio. And how are we able to get it? You can see, you just see the mathematical interpretation along with the result. 3 is to 1 monohybrid cross result. 3 tall, 1 dwarf. Here 3 is to 1 round wrinkled for tall, as well as 3 is to 1 round wrinkled for dwarf. For each category, 3 is to 1 yellow green for round tall, yellow green 3 is to 1 wrinkled tall, yellow green 3 is to 1 round dwarf, yellow green for wrinkled dwarf. So here you can see all the 3 are dominant, tall, round, yellow. So 3 into 3 into 3 we get 27. Second one, tall, round, green. 2 dominant, 1 recessive. So 3 into 3 into 1, 9. Again here you see 3 into 1 into 3, 9. Again you see 3 into 1 into 1, 3. Very clear? So mathematically you are able to find the solution or find the ratio without going to the garden for experiment. Mendel did it. Okay, and here dwarf 1 into 3 round dominant with 3 yellow. So double dominant, 1 recessive. 1 into 3 into 3 9. And here dwarf round green, 1 into 3 into 1. In the same way, dwarf, wrinkled, both are recessive, one dominant, 1 into 1 into 3. And finally, all the three are recessive. So 1 into 1 into 1, 1. So this is the 27 is to 9 is to 9 is to 9 is to 3 is to 3 is to 3 is to 1 ratio, students. Understood? The mathematical interpretation of Mendel's Trihybrid cross ratio. Okay, so this is the end of this video, student. In this video we learned the binomial expression based on the monohybrid cross, Mendel's work, why it was not recognized, reasons, rediscovery of Mendel's work, then chromosomal theory of inheritance, work of Morgan and his colleagues, then from the state board book Trihybrid cross. These are the concepts we covered. Thank you very much for listening.